All right, you guys, welcome back. Today we're going to talk again, chapter 2 about cells, but this time we're going to focus on part 2, which looks at the cell cycle. So without further ado, let's get going, shall we? All right, so what is the cell cycle? Well, first of all, it's oftentimes known as cell division, and that's simply when one cell grows, is big, then divides into two. And this is actually a very important process because the cell cycle makes sure that there's precise replication of the DNA. So you have an extra copy of it. And then it equally distributes the DNA to opposite sides of the cell so that when the division occurs, you end up with two identical daughter cells. They are identical to each other because they have the original information or the same information, and they're identical to the original cell. So if you look at this picture, this is a chromosome, and one side of it would be the original, the other side would be a copy of it. So it's two copies of the same information. So you have two big chromosomes, one pink, one green, two small, one pink, one green, and we end up with two cells, each with one pink, one green big, and one pink, one green small, just like we started off with. All we did was we separated the copied information and then move them to opposite ends of the cell and divide this cell into two identical versions. So let's continue talking about this. I mentioned chromosomes. Chromosomes are coiled chromatin wrapped around histone protein. So what we've done is we've taken the entire DNA, all the chromatin, and we've kind of chunked it and wrapped it into these chromosome forms. At the center part is something called a centromere. And this has particular proteins that allows the duplicated portion, also known as a sister chromatid, to attach to it. So if we look here, these are the original chromosomes, but then we duplicate our information. So I take this yellow information and I duplicate it. They are attached here in the middle. That is the centromere, and these are my sister chromatids. Then I take the blue information and again, I duplicate it. So I have one copy of it connected via the centromere to another copy of it. These are the sister chromatids. The telomeres are the very, very tips of the chromosome, so the top tips and the bottom tips. Now, all together, these make up homologous pairs. Now, homologous pairs are simply two of the same chromosomes. Remember, we said that your chromosomes come in pairs. You have 23 pairs of chromosomes. So you have two of every chromosome, and you got one from mom and one from dad. Now, it's the same chromosome that you got from mom and dad. They'll have the same genes on them. They might have different variations, different alleles for those genes, but it has the same genes. And that's what makes up homologous pairs. Those are homologous chromosomes. So these are homologous chromosomes, and then we duplicate them. They're still considered homologous chromosomes. Diploid is a very important term to know. It is when a cell contains both sets of chromosomes. In other words, it has the full set. For humans, that means that every cell that is diploid has 23 pairs of chromosomes. So it has all 46, as opposed to haploid, which is half the chromosomes of diploid. So a diploid cell would have both the yellow and the blue in it because it has the pairs. A haploid cell would either have the yellow or it would have a blue, but it would not have both of them. So it only has one of each chromosome. There are two main classifications of cells. We have somatic cells and then gametes. Now, gametes are also known as sex cells. Somatic cells are your body cells. So these are pretty much any cells that are not your gametes. So your skin cell, your bone cells, your lung cells, heart cells, brain cells, blood cells, etc. All of those are considered somatic cells. And they are all diploid, meaning that they have the pairs of chromosomes. So that means we have 46 chromosomes in all of our somatic cells, 23 pairs of chromosomes in all of our somatic cells. But our gametes, or our sex cells, are haploid. Now, these are the eggs and sperm. And what it means to be haploid, again, it means you have, instead of pairs of chromosomes, you only have one of each chromosome. So that means instead of having 23 pairs or 46 total, your sex cells, whether they are sperm or eggs, would only have 23 chromosomes, one of each. And that's what this picture below is showing you. So here we have an example of being diploid. We have 3 homologous pairs. That's a homologous pair, that's a homologous pair, and that's a homologous pair. So we have 3 homologous pairs. We have they come 2 for each one. So the N stands for number of type of chromosomes, and 2N implies that there are pairs, 2 of each. So we have 1 pair, 2 pair, 3 pairs. So 3 is our N number. 2 times 3 gives us 6. That's how many total chromosomes. 1, 2, 3, 4, 5, 6. So a lot of times we talk about diploid as being 2N, 2 times the number of types of chromosomes. Haploid is just N because there are no pairs. So we have this chromosome, we have this chromosome, we have this chromosome. No pairs, just 1 copy of each one. 1, 2, 3. That was our N number. And if you notice, or if you remember, that was our N number over here too. So this has half of these. Notice we see this guy, see the blue one, and we see the green one. They don't have the pairs anymore. All right, so what controls it? Well, there's actually both external and internal cues that will control when a cell will divide. So each type of cell has their own set of cues. For instance, skin cells, they have cues that tell them to divide very frequently. In fact, your entire top layer of skin is already dead. It doesn't get sloughed off and gotten rid of until the previous or the underlying layer goes through mitosis, and then you have a new dead layer for the top. Liver cells can do some wound repair recall, meaning that they can repair damage up to a certain point. Nerve and brain cells really don't go through division at all. So once they are formed and functional, they really don't change that much. So that's why nerve damage and brain damage can be so severe. I mean, you cut your skin, it'll heal. It'll leave a scar, but it'll heal. You cut a nerve and you've lost function with that nerve. It will not work again. G naught, so that's supposed to be a G with a subscript of 0, stands for G naught. And your G naught phase is when a cell might switch to a non-dividing state. So it kind of takes itself out of the cell cycle for any number of reasons. But first, let's talk about the cues. Let's first talk about external cues. And the biggest one of this is density-dependent inhibition. So what that means is, remember, we talked about how the extracellular matrix interacts with the different cells and allows them to stick together and recognize each other. This also allows for something called density-dependent inhibition. So your skin cells, they stop dividing once they reach each other. So you have this nice layer of skin. However, if I get a cut on my skin, there's now a break in the cells. And the extracellular matrix and the carbohydrate chains hanging off of the cells recognize that they are no longer touching another cell on one side. Since they recognize that, it's an external cue that's going to cause them to start preparing for cell division. So they'll go through all the preps of cell division, they will divide, and they will keep dividing until that wound is completely sealed, until each cell recognizes that it is completely surrounded again by its fellow skin cells, at which point the cue turns off and they switch back to just doing their regular skin cell thing. Now, tumor cells actually typically lack this inhibition, which is why they constantly will keep growing. They'll keep dividing and dividing and dividing. It's simply because they're not told, all right, you're touching more cells, stop. That cue is always on. Internal cues. So there's different cues that tell the cell when it should regularly divide on its particular schedule. Now, we mentioned that skin cells divide frequently. So it has these internal chemical-based cues that keep it on a schedule. And what it's based on is these cyclin kinase pairs. These are the amount of these, relative amount, will actually affect the genes, turn certain genes on, which will then trigger the activation of mitosis. Mitosis is simply cell division. So then as mitosis begins, then you have the enzyme kinase breaking down cyclin. So pretty much what's going on is you have this molecule called cyclin. It gets made and the concentration of it builds up. When it reaches a certain level, that activates certain genes, it turns them on to create something called kinase, which is an enzyme. Now, the creation of kinase triggers mitosis to start. So it's going to start going through cell division. But as it's doing that, the kinase is going to start breaking down the cyclin, so that concentration is going to get low. By the time mitosis is done, the cyclin levels are low once again, and then they have to be built up. So then they're going to start, they switch the genes for the kinase off, and we start building up cyclin again. It takes a certain amount of time. Once you get to a certain amount of time and a certain amount of cyclin has been made, activates the kinase genes. Kinase is created to break it down. This causes mitosis to occur, and so on, so on. Cells also have an internal clock that tells them how many times they're able to divide, and that's based on something called the telomeres. Now, we said that telomeres are the ends of the chromosomes, the tips of them, and every time you go through DNA replication, you actually lose a piece of the telomeres. So they shorten over time. And once they get to a certain point where you lose the telomeres completely, the cell is actually no longer able to divide because it can no longer make copies of its own DNA. So at that point, the cell cannot make copies of its DNA to put into new cells, so we just stop making new cells. And we do see that kind of occur as we age. All right, let's talk about the cell cycle. So there are two main parts. The first one is called interphase, and this is the part that the cell spends most of its time in. And then there is M phase. M phase is when it's going through division, also known as mitosis, and there's something else called cytokinesis that occurs at the end of mitosis. So interphase may last several days. For instance, if you are a skin cell, it might last several days. It could last years. So there are certain cells that will go years before they divide again. And even brain and nerve cells, they really don't divide a whole lot once they are formed. Now, can you make additional neurons and all that? Yeah, you can definitely make new neural connections. But again, there's going to be years between a lot of those divisions. And during interphase, that's just simply the cell doing its job. Whatever kind of cell it is doing that job, which we'll talk about when we get to discuss interphase in more detail. Mitosis is when it pauses doing its job, goes through the steps of division, and then you end up with two identical cells, identical daughter cells, and they both go back to interphase. All right, so As you can see from this picture at the bottom, this entire bit is interphase, and just this little bit is M phase. So once we have a new cell from the previous division, it goes into G1 phase. This is a growth phase. So what happens is, since it just came out of a previous division, it's pretty small. So the cell is going to grow. It's going to double in size. It's going to get bigger. And there's also a restriction point in this. So it actually does a self-check. Are the organelles made properly? Is the DNA made properly? Does everything look as it should from the previous division? If so, it keeps going. If not, if it's a no-go, then it's actually going to enter G naught, that G with the subscript of 0. You'll notice that right here. So it self-checks itself, make sure everything is fine. If it is, it keeps on going from G1 to S phase. We'll get to that in a moment. If it's not fine, it goes to G naught. Again, we'll talk about that now since we brought it up, but the information is at the bottom here. So this is a non-dividing state. When a cell enters G naught, whether it's because it's out of telomeres or because it recognized something was wrong from the previous division, it'll go to this non-dividing state. So it removes itself from the cell cycle. If it is because something is wrong with the cell, it will try to fix it. If it's able to fix it and it does another recheck, everything turns up fine, then the cell can actually rejoin the cell cycle. It can go back to G1 and continue on. If it cannot be fixed, or if it has no more telomeres and essentially the cell can no longer divide, then it's going to stay in G naught until something called apoptosis, which is programmed cell death. We'll talk about that in a little bit. Now, not just growing occurs in G1. It's also going to start making a lot of proteins. So depending on the type of cell it is, it's going to make different types of proteins. Certain skin cells are going to make pigments. Pigments are proteins. Now, whether they are pigments for the skin or they're at the base of your hair follicles and they make the keratin, which is the protein that is hair, as well as pigments to go with that, or if they make keratin for your nails, that's going to be different than the proteins that your stomach cell makes or that your brain cell makes. So it's going to be making proteins and doing its normal job. Whatever kind of cell it is, it's going to be making proteins to do the job of that cell. When it gets a trigger saying that it should start getting ready for the possibility of mitosis, it switches from G1 to S phase. Now, S phase is when it will replicate its DNA. So it's going to take its DNA, it's going to separate the double helix, and it's going to read each strand and make copies so that you end up with 2 copies of your entire genome present within that cell. Once S phase is completed, it goes to G2 phase, which is our second growth. So the cell's going to continue growing. It's going to make sure it gathers extra material for the division. It's going to do final preps for division. It's going to make the enzymes that it needs for divisions. It's going to make extra copies of the organelles for the daughter cells of division. So it's just going to keep growing and getting ready to divide. And then, once it's all done with that, that's when it moves to M phase, which we said is mitosis and cytokinesis. Now, mitosis is broken up into 4 parts. A lot of kids remember it using the acronym PMAT, P-M-A-T. And that stands for the 4 main parts of mitosis. We have prophase, metaphase, anaphase, and telophase. Now, there is like a pro-metaphase. We're not going to worry about the in-between ones. We're just going to worry about these 4 main ones. So during interphase, we duplicated our DNA already. That was in the S phase of interphase. In prophase, the chromosomes become visible, meaning that the DNA switches from the chromatin format to the chromosome format. The centrioles, we talked about those last time. They're an organelle that only functions during cell division. They're actually going to start separating. So that's what these little stars are. They're going to start moving to opposite ends of the cell. The nuclear membrane is going to start to disappear. So temporarily, the chromosomes are no longer housed in the safety of the nucleus. It's going to disappear temporarily. As the centrioles are also moving, they're going to start building these things called spindle fibers. Now here we have the next phase, which is metaphase. So those spindle fibers that were formed from the centrioles, they're going to attach the chromosomes, attach to the chromosomes at the centromere. So it's going to attach at the centromere, and then the spindle fibers are going to line the chromosomes up along the middle of the cell, as you see here. Now we're going to switch to anaphase. The centrioles pull on the spindle fibers, causing the spindle fibers to pull on the chromosomes. Now, when they pull on the chromosomes, it's going to cause the centromere to split and separating the sister chromatids. It'll continue pulling and start pulling those sister chromatids to opposite ends of the cell. During telophase, the spindle fibers break down, the chromosomes disperse, meaning that they're going to go back from chromosome back to the chromatin form. The nucleus is also going to reform around them. At this point, during telophase, this is when cytokinesis occurs. So cytokinesis refers to the division of the rest of the cell. Mitosis looks at the chromosomal separation. Cytokinesis looks at the division of everything else during the cell. So the cell membrane will start to pinch in, like you see here. And as it pinches in, it causes the cytoplasm to stream to each end so that each end is filled with cytoplasm. It's also going to cause the organelles to be separated and move to each end as well. Eventually, the cell membrane will pinch so far in that it fuses, and then it'll pop apart, and we are left with two identical daughter cells. All right, so let's take a picture, or take a look at some pictures. So first, mitosis 1, it's a comparison of real images versus diagrams. So these are actual images taken using a microscope. So early prophase versus late prophase. The nuclear membrane is breaking down, the centrioles are starting to move, and they're also starting to build their spindle fibers. The chromosomes become visible because they're switching from chromatin to chromosome form. The spindle fibers start to attach to the chromosomes at the centromere. Metaphase, the spindle fibers are all attached to all the chromosomes, and they line them up along the middle of the cell. And still see how they're in a clump. And then here, they're lined up, more or less in a straight line. These clouded regions here, that's where the centrioles would be. Anaphase, the centrioles pull on the spindle fibers, which pull on the chromosomes, causing the centromere to split and separating the sister chromatids. It'll pull the sister chromatids to opposite ends. So you see we have two clumps being pulled away from each other. And then telophase, the chromosomes go back into chromatin form, the nuclear membrane reforms, the spindle fibers break down, and then cytokinesis occurs at this same point. The cell membrane starts to pinch in, causing the cytoplasm and organelles to stream to both ends. So notice this. Chromosomes aren't as visible here, and we can also see the pinching in of cytokinesis. All right, again, just another full diagram. So we start off with the cell. It goes through interphase, which is when it's going to duplicate and replicate its DNA. Now the difference between this picture and here, this would be G1. This would be G2. So we start off smaller, it gets bigger, replicates its DNA in S phase, then we're in G2, where it's gathering material. It's even made extra organelles, like an extra set of centrioles. Now we switch to M phase. So we go to prophase first, nuclear membrane disappears, chromosomes become visible, centrioles begin to move, and they're starting to form their spindle fibers. Metaphase, the centrioles, their centrioles are on opposite ends. Their spindle fibers have attached to the centromere of the chromosomes and line them up in the center of the cell. Anaphase, the sister chromatids are split and dragged to opposite ends of the cell. Telophase, the nuclear membrane starts to reform around the chromosomes. The chromosomes start to switch back to chromatin form. Spindle fibers break down. And cytokinesis, the membrane starts to pinch in on both sides. It's going to cause the cytoplasmic organelles to stream to opposite ends until it's pinched all the way through, fusing and separating to form 2 new daughter cells ready for G1 of interphase. And then this is just a picture we saw before, kind of showing an idea of time, how the majority of time a cell has is in interphase. And then mitosis is going to be a very, very short amount of time. So this can last anywhere from days to even years. This is going to be over in a matter of hours. All right. Additional resources. Now we're not going to watch any of these videos during this presentation. I will post a link to this whole Prezi in the video description so you can access it and access these videos. So Science in Pajamas, this is a series I've created on YouTube that talks about different scientific stuff. We kind of go over it, explain it, use diagrams or other examples. So first we have one on prokaryotic versus eukaryotic cells. We have one on animal cell organelles, so identifying them, as well as a little bit about their functions. I know we didn't really cover plant cell organelles as much, but most of the cell organelles are the same between the two. So if you want to kind of review that a bit as well, you could. Cell organelles as well as their function. And then we have one on the cell cycle. I also have other videos of different sources that can help you. So there's an amoeba sisters prokaryotic versus eukaryotic cells. I know it looks all silly and cartoony, but the amoeba sister videos are amazing. They very visually interesting, and they also do a great job of breaking down and summarizing the information. Amoeba sisters again, introduction to cells, the grand tour. Bozeman science, a tour of the cell. Amoeba sisters, mitosis, so again, talking about division. Amoeba sisters, the cell cycle, and this one also kind of deals with what happens when the cell cycle goes wrong. And then Khan Academy phases. Ooh, I will make sure that that is actually posted. There will be a link to the Khan Academy phases of mitosis video. I'm not quite sure why it's not there. I could have sworn I put it in. Maybe I didn't save it. All right, so that's it for this. If you have questions, hit me up in our Google Classroom or email or actually in class. But I will fix that Khan Academy link so the video is there. And this way, when you click on the link for the Prezi, you'll have access to it. But in the meantime, you guys just stay awesome, stay amazing, stay curious, and stay safe and healthy. All right, you guys, until next time. Bye-bye.